for today, for the week, for whatever, is Eran Halperin. Please, Eran.
Okay. Can you hear me? Yeah. All right, thank you. So I'm going to try to, so I have a bunch of things planned, but we're a really small group. I prepared this slide, but Michal already kind of ruined the surprise. but this is, that's the Kaplan-Meier for the number of people that survived since the beginning of the week. So a few things, just kind of to set the stage for the talk. I kind of thought about basically theory and healthcare, and I wanted to kind of see if I can bridge some of these things and talk a little bit about things that could be interesting for the theoreticians and for the physicians. I don't know if we have any physicians left in the room, but maybe the biologists. So before we go, there's all the, including this one, all the images are either AI generated, or I took the pictures, or I give credit. So this is the disclaimer. So I'll start with the first one here. I just, I think I mentioned this in the beginning of the week, that I started from theoretical computer science, and some people like Omer and others maybe know me. And I used to work, I spent many years here, and I used to work a lot with Dick Karp, whose picture is on the other side. Not related to the talk, but it is related in the sense that what I really love about theory and all the talks that we heard today and this week is that it's super elegant and very clean. Not as messy as when you go to healthcare. Healthcare is completely the other side. I mean, it's very impactful. Obviously, if you make every small thing that you make, every small improvement, it has an impact. But it's very difficult to do essentially what Matthew did or Omer or any of the, in terms of kind of formalizing it in a way that is going to be very clean and elegant. Personally, I love both worlds, but I'm going to try to show some things that at least would, maybe for the theoreticians here in the audience, will help you or maybe trigger you to think about some of the problems. So I have two concrete ones. One I'm going to mention in the middle, and if I have time, just at the end, something which is a little bit more kind of vague, but I think it's important. If not, if we won't have time, I'll just do it offline. So this, so when I'm thinking about healthcare, it's messy. Some of it, Ron Ballister gave in the first day, gave an excellent talk that basically talked about all the misaligned incentives and so on. And I think this is a critical thing that you have in healthcare. When we had these talks over the week, I missed a few talks, but generally what I saw on the healthcare side was either kind of a discovery or I would say life sciences side of things, or public health. But I just want to point out that there's way more than that in terms of when you think about healthcare. Anything that is essentially related to kind of understanding the relation between all of these groups is within the healthcare, and there's a lot of things. And I personally spent a few years in United Health Group in Optum, so and a few of the things that we just discussed basically were there. When we think about these misalignments, I think, the other thing is, I want to try to make it interactive, and some of it is going to be a little bit, I'm going to ask you some questions. Some of the questions are not going to be scientific, and specifically, when I think about misalignments, I was trying to think about what examples, kind of in the general media there are for misalignment. So, question for the audience. These are coming from three different kind of entertainment things that are related to healthcare misalignment. Any guesses, or nobody watched any of these? Okay, so Theranos, people heard about Theranos? Okay, the Dropout. Okay, so you don't have to watch the Dropout, you can read Bad Blood, but basically Theranos is one. So Theranos, the idea was it's a startup that basically tried to have one sample, one blood sample, or actually there was no blood in principle, that is going to tell you everything. And I'm kind of going to connect it later when I talk about methylation. I don't promise to be Theranos, but basically there's something similar about it in the sense that it tells you a lot of things. Painkiller, nobody watched Painkiller? On the opioid addiction, a very good one, so I highly recommend. So this is on the opioid addiction and the misalignment over there between the pharma and the providers and so on. And then older one, it's Gattaca. So Gattaca is fictional, but basically the idea over there is that you can say, so that's a movie from, I guess the 90s, I don't know. Even longer, yeah. But the basic idea over there is that someone is born, or when the baby is born, you can tell that that baby is going to get a heart attack when they're 30 years old exactly. And then the whole, instead of an interview, you take a hair of someone, you get their DNA and things like this. So these are all kind of pointing out like the issues that are the complexities of healthcare that are that you can. That we have to tackle, not beyond just fairness. Basically, I think there's a lot of issues here that we have to think about. But one of the things that if you just go back to the Gattaca example, that I think it's clear to all of us that is going to be useful, and we talked about it, a few talks basically look at this, is how can we use AI to do disease prediction? I mean, Matthew just basically mentioned the foundation models for this, but in general, even if we go back to the first day when we talked about, there was in Rand and Noah's talk, there were, I forget how many people, but basically 50,000 people that they prioritized, they got to 40 or 50. That is essentially a model that tries to predict who is going to be, to have a problem, and then based on that you prioritize who you're going to call, who you're going to give treatment, who you're going to give resources, essentially. So that's a general problem in healthcare, and by the way, this is related both to the things that Ron was talking about, so both for the providers and for the payers in the, let's say, in the US system. So both of them are basically trying to figure out who's going to be sick tomorrow, because what can we do about it if there's something to reduce cost, to help the health of the patients and so on. So I'm just quickly going to give just to mention a few things that we've done. I'm not going to go into those, but just to give you kind of an examples beyond just the EHR. I'm going to talk in 2 slides about the EHR. Matthew basically kind of talked about it, so I'm going to go very quickly. But so this is just an example for within. When you have a surgery, or if you're in the ICU and you're a high-risk patient, then they would measure your blood pressure continuously, because this is it basically tells you if someone is going to have, or at least it's a kind of a predisposition for risk after surgery. So what we did here is we tried to do the same thing without the invasive part. So essentially looking at ECG and pulse oximetry, so basically the PPG, and use, that's a VNet, I don't know if you can see here, but basically use neural network to get the continuous blood pressure. So this is one example. So it's not really predicting a disease, and it's actually not predicting the future. It's predicting something that happens at the same time that we know that is related, that it's a risk factor for something that is going to happen in the future. And that actually is pretty common. That kind of a common task is to know what the patient has today, because this is affecting how we think about the patient later. That's another example from a different field. So this is from ophthalmology, where we have a series of models that are, we don't call them foundational because they're not, but they are trying, the basic idea over there is to look at, it's very difficult to get labeled data, and these are 3-dimensional data of the retina. So kind of volumes of the retina, and you're trying to find these biomarkers that tell you if someone, this is age-related macular degeneration, which is a very common eye disease. And you're trying to see if the patient has these biomarkers today, because that is a risk factor for what's going to happen to the patient later. So again, these are all not predicting what's going to be in the future. It's predicting the now, But from the literature, we already know, the clinicians think about it all the time, what are the risk factors that are going to affect us later. So even just predicting what's going on now, when we have a patient that comes into the clinic, definitely in the US, we don't know much because they come from a different system. Maybe in other countries it's sometimes a little bit better, but just generally you don't, in 8 minutes you don't really know a lot. And the question is, what can you do? What can you tell about the patient now that in a kind of automatic way, or just how can you enhance the information that the clinician has? Okay. We just talked about it, so I'm just going to say quickly. So we built this kind of a foundation model. It's basically a GPT it came out in 2023. So we actually worked about it on this before ChatGPT came out. And when ChatGPT came out, I guess it was kind of like, okay, it's clear that everybody should learn. And we're not the first ones that did that. We were the first ones that used the generative model. Before that, there were a couple of papers that used BERT, which is not generative. So this was done in my work in Optum, and actually we did use it to predict, essentially kind of like similar to what was mentioned earlier in the week, which is you're trying to prioritize which patients you're going to call based on the risk for a finite set of things that you're interested in. And that's where I think it was a little bit different than what you mentioned, Matthew, because we had maybe 100 different things that we wanted to optimize. Some of them were more rare, and then that was actually where we got the boost. But in principle, if you had something like this that worked really well and efficient, then you could also simulate more data. Then you can democratize EHR data across the community, right? Which I think would be nice. Now, one of the problems that I think we mentioned about EHR, but if we didn't, let me just repeat, emphasize this, is that EHR data is, first it's sparse and it's incomplete, but it's also biased, right? So think about it, when you go to the physician, you may have an ICD code, a diagnosis code that is missing because a test was not done for you, or because they don't have the history of that, because it's a new system. And you may have an ICD code, a diagnosis code that is there, but you actually don't have the condition. And the reason may be because they want to put it for billing, because they want to do a test. And in order to do the test, they have to tell the insurance company that they need that you have the ICD code, they have this diagnosis. You see it all the time. Just as a kind of a simple example, if you look at rare conditions that are supposed to be in 1 in 10,000, you'll look at the EHR data and you'll see them in 1 in 1,000. Right, so 10 times more than what you expect in the population. And that's, for many conditions, you'll see this. This is obviously a bias, right? So this is something, so the data is not clean, and it's not only that it's missing and it's biased, you have overrepresentation and underrepresentation depending on what, which makes everything that we're doing, including basically what I just showed you before, the GPT or any foundation model, it has to deal with it, and we don't really explicitly deal with it typically. So that basically brings us to the question of, can we impute the data? So can we impute these unknowns? There's a lot of imputation methods that would take you can think about, like, you have a matrix. This is the kind of a EHR matrix for the patient, and you do some matrix completion method. There's many methods that are doing this. Most of these methods, or the typical methods that are used, are not taking into account the fact that you have missing, not at random, and these bias problems, right? So they don't assume that someone intentionally add noise in an adversarial way. That's one thing. And so, but we have done this in the past. We've used these kind of methods in the past when we looked at these type of models and others. And we started asking the question, can we use imputation from genomic data? And the first question is, can we do it from genetic data? So looking at, let's say, polygenic risk scores. So just quickly for the non-geneticist, and I don't know if we went over this in one of the talks that I don't know, Michal, did you go over this? No, I'm not sure. Okay, I'll just do it very quickly. But basically, you have, if you look at the genome, you have a lot of these variants. We call them, so let's say the most common ones are called SNPs. These are just differences in specific positions. And so each of these, let's say each of these rows is going to be one individual, and you'll have these different variants, and different people will have variants in different places. And then you do this kind of a linear model, and typically with weights. Just here for the sake of simplicity of what I'm writing here, there's no weights, but basically it's a linear model that takes into account all the variants. It just sums up the variants that you have over there. And then you have this kind of, because you just sum up over many, many things, you have a normal distribution, and you're looking at these kind of people that are at higher risk, and you hope that you can say something about them that is interesting. So there's, I just put four references here, but there's thousands, essentially, of references of people basically trying to optimize this PRS and trying to show utility of the PRS and things like this. So this is all done with genetic data. With these common SNPs that you have, depends how you count, but let's say millions of those out of the billions of base pairs that you have in the genome. And then, but it had some limitations. So let me just go quickly over these three bullet points here. So the first thing is genetics is genetics. It doesn't capture the environment. And that means that if you can, so this is basically one thing. If you smoke, if you don't smoke, it doesn't really matter. Your genetic is going to stay. Second thing is that genetic is static. So going back to the example of Gattaca, if I can tell you today that you're going to have a heart attack next year, that means if I can tell you this from the genetics, that means I could have told you this when you were born. Now, there could be some conditions that when you're born, you could basically tell a person exactly when they're going to die or when they're going to get a specific disease. But intuitively, at least, it's not really clear that it's true for most conditions. Again, we don't know, but that would be my intuition. And so that's a huge limitation if you're looking for the short-term risk, not lifetime risk, but just a short-term risk. Now, the other thing is that, PRS, I showed you the first papers came out, almost 20 years ago, I guess. I don't know that they're used in the medical community at all. I don't know if but there's talks about it. There's definitely a lot of pushback from different sides, and maybe it's because of the misalignment. But I don't want to say exactly why, but it's not there yet. It's definitely not a common thing that is used all the time. And for 20 years of a lot of research that is going into this, it's kind of it's either because someone really doesn't want it, or because maybe we need to do more work to show that it's useful. Yeah.
Yeah, I think that the problem that we didn't know very recently, 2010, that we had this SNP GWAS top X that were explained very, very low of the variance, very small. But then we realized that all the message, that, for example, for height, 62% of your SNPs have a no-zero effect.
Yeah.
So it's really like an infinite model. So the more you will have patients, the more you will have information, for sure. But on the specific question, so that will be another aspect. So yes, there will be some information there. It will come, but we need, it doesn't.
So there's many potential. Yeah. So that could be a reason. There are many some reason. I think I forget if it was Michal that mentioned like interactions or but potentially there's interactions that we don't take into account here. So maybe there's more information. And by the way, when we look at heritability, we also don't look at interaction typically. Unless you do something else. But normally you don't look at interactions. And so there maybe all the information is in the genetics. We just don't know how to capture it now. Maybe when we're going to have 70 billion genomes, then we'll know.
Really the message that it's expected that we have very low probability with the PGS now, but we will have more. But again, it's great to have other properties.
Yeah.
Because,
Just to say, if you look at 50 PRS done with the best, across ethnic, you name it, the last 2% are often, on average, I mean, there are cases, risk factor of 4. And
Okay.
risk factor of 4, if you have wine a day, it's risk factor of 8. So the signal is still very low.
Yeah.
So I mean, I wouldn't want to generalize. Maybe in some cases it is
Some
there,
cases it is,
but then you have to ask the question: is it more there than basically just asking for the family history, for example? I mean, there's a lot of questions.
Yeah. No, it's an excellent topic.
I'll tell you the pushback that I heard from, I cannot say, I'm in video, I cannot say who, but basically, the pushback is that, okay, the information in the EHR plus family history and so on is I mean, even if there is information here, they don't see that there's more information when you go. At least that could change. But the thing is, it has been 20 years, roughly 15, 20 years, and we're still not there. I don't know the reason, and there could be scientific reasons, and there could be non-scientific reasons. These two things are, I think, these are facts, right? So these are limitations. So one of the things that we kind of try to think is, can we use a methylation actually for this? And we haven't, I think, Elior he gave a really nice talk about methylation today, but he didn't really define methylation. I wasn't sure if the theoreticians know what methylation is. So just kind of in general, no? Okay. So, we have the DNA, and the DNA is fixed across all, almost fixed in all our cells, almost the same. But on the DNA, there's changes, there's modifications. So basically, other kind of molecules that are attached to different parts of the DNA. And so in this case, methylation, these are methyl groups, but it doesn't really matter. These are basically another molecule that is going to every specific DNA, so it will be different between different cells. Methylation has been around. People know about it for a long time. From mathematical, theoretical, or for the computer scientists here that don't have the background, basically it's really just a matrix of data that the numbers are, if you think about genetics, it's going to be 0, 1, 2, and here it's just numbers between 0 and 1. And you have individuals and you have these methylation sites, we call them CpG sites. Doesn't really matter why the name is CpG, but so the columns here are going to be the CpG sites. Let's say that there's 800,000 of those columns, typically in a typical experiment, and the number of individuals depends on your sample size. So that's methylation, and I was very intrigued by methylation when basically I started kind of learning about this, which was maybe about a little bit more than 10 years ago. People learned about it before me, but this is where I started getting interested in. There's a couple of papers that came out around the same time, 2013. On the right side, you see the DNA and methylation age. So it's called a methylation clock. So the methylation clock basically says that I can take a blood sample from you. Actually, I can take any tissue. This paper shows that I can go to any tissue in your body. I don't know if any, but Steve Horvath that did this work took many different tissues, and he showed that there is a signature in each of the tissues that tells you your age. And I can go to each of the, so I would take your blood sample and I can tell you your age roughly up to maybe 1 or 2 years of error. And we tried it also, so we never published it, but we implemented, or we developed a methylation clock for the data that Elior showed this morning on the children with asthma. And over there we could get 2 or 3 months error, because these are children, it's like the ranges. So it's very, very accurate, which essentially means that there's methylation sites that how much methylation you have in your blood, or actually it's true for other tissues again, is going to be highly correlated to your age. And there's a lot of literature about like getting better clocks and things like that. But this seemed to me like a very exciting thing in the sense that this is almost like magic, right? This is so strong. It basically captures something that DNA, genetics, can never capture. Genetics cannot tell us how old we are because it's static, right? The other thing that I was very interested in is this paper that Elior also mentioned. This paper. This is a paper that showed that there's a lot of relations. Essentially, the entire genome is correlated between rheumatoid arthritis and methylation. So they kind of the paper basically, the title says, okay, methylation stands somewhere between genetics and the disease. So maybe we should learn, we should do, instead of these GWAS, genome-wide association studies, where we're looking at different SNPs that are related to the disease, maybe now we're going to look at methylation sites that are related to the disease. So like from that analysis, basically, essentially almost every methylation site in the genome was related. And then, so a lot of this work is actually with Elior that I'm going to mention now. What you see here is, Elior mentioned it in his talk this morning. Someone asked about, I think maybe you asked about like the population. So what you see here is, I mean, you have to do some tweaking. It's not really PC1 and PC2, and I'll explain in a minute exactly what it is. It's PC1 and PC2 after you do something. But these are two populations. I believe these are Mexicans and Puerto Ricans. And this is the methylation data of these two populations. You see that they're separated. So it captures essentially, if you think about it, that's really the genetics, because the genetics is what defines populations in that sense. Again, going back to GWAS, EWAS, so GWAS is genome-wide association study. When we're looking at SNPs, people have started basically when they saw that methylation has so much promise, they started doing these EWAS, epigenome-wide association studies, where we're looking for a methylation site that is correlated with the disease. So it's exactly what we discussed this week a few times. You're basically going site by site, and you're asking, is this site correlated with the disease? You do a simple regression, and you're trying to find the ones that have good p-value. Now, if you don't correct for anything, this is what you get when you get a QQ plot. I'm not sure if everybody is familiar with the QQ plot, but the basic idea with the QQ plot, quantile plots, you look at the expected p-value. You expect p-value should be uniformly distributed. So you look at the expected p-values, and you look at the observed p-values, and it's crazy inflated. I mean, you don't see these things except in, I don't think I've ever seen, since then, I've ever seen anything that inflated. Now, it could be real. I mean, think about it, 2013 could be real in the sense that maybe everything is related to the to rheumatoid arthritis. Maybe it's true. But we wanted to see how can this potentially be explained. So one of the things we did is, and this is what you see here is essentially kind of similar to quantile in the sense that I'm looking at a random matrix. If I look at the random matrix, I just generate a random matrix, let's say with normal distribution for every entry, and I look at some matrix X, I look at the X transpose, and I'm looking at the eigenvalues of this matrix. There is a distribution that is well known. It's called Marchenko-Pastor of how the eigenvalues should behave. And this is, again, the observed compared to the expected, or expected versus the observed. It's not written here, sorry. But one of them is expected, the other one is observed. You can see that it's inflated. What does it mean? It means that you have a lot of eigenvalues that are way bigger than what you expect by chance, which essentially means, what is an eigenvalue? Eigenvalue is a direction that the entire matrix basically goes in that direction. So it's one of the, you have a linear transformation basically that a lot of, you have a lot of points that are going, a lot of samples that are going in that direction. So typically a confounder will give you these kind of high eigenvalues. And what you see here is essentially a huge amount of confounders. Typically, if you look at, let's say, genetic data, if you did the same thing, you'll get something which is straight, and maybe 4 or 5 points. 2 or 3 of them would be related to ancestry and maybe 1 or 2 to batch effects or something like that, right? Technical effects. Here you see essentially half of it is basically confounders. Now, some of the confounders are technical. They're not real, they're not biological, and some of them may be biological. So one that is well known for methylation and other genomic data of this sort is that you have kind of a typical pattern for different cell types. So let's say that you have 2 types of cells, blue and red. And let's say that in one of the cell types, in the blue cell types, they're always methylated, and the red cell types, they're never methylated. Then there's going to be a perfect correlation between whether it's blue and how much it's methylated. Right? So you'll have 60% here methylated, but also 60% blue. What you really found here is that there is 60%. So the fact that you have a difference between the cases and the controls is because you have 60% blue cell types versus 40% red cell types, and not because of anything that is specific to that methylation site. So it's a biological confounder. Age, by the way, would also be a biological confounder, right? Sex would also be a biological confounder in methylation, not in genetics. But you can actually see that there's a lot of differences between methylation between men and women and many others. So this specific one is something that people know about and they try to correct, including they try to correct it in the rheumatoid arthritis paper that I showed you before. If you look at, so one way to correct this is using some variation of PCA. This picture is from a paper, kind of a classical paper in statistical genetics, where the idea was you take now genetic data, so DNA, I forget how many people were here, but maybe a few hundred, and you run PCA. You take the first 2 PCs, and you just plot them on the kind of a PC1, PC2 map, and you color them based on the ancestors of these people. So for every person, these are people from Switzerland, but they asked them where they took people with 4 grandparents from Italy, 4 grandparents from Spain, so they're always from a specific country. And you kind of see that what you get here mirrors the colors here, and you just get the map of Europe, roughly. And that basically means that the first 2 PCs capture that information of ancestry, that confounder. Yeah.
Yeah, this is the ancestries of the 4 grandparents. That's super important because some people are using this to say that I am a proto-French. If I go to Paris, it's much more.
Because people will be mixed, you mean?
Yeah.
Yeah.
So it's very important because that's, for example, UK Biobank, they really make. They were super clear that they want the ethnic population of UK, not the UK from 200 years ago.
Right.
That's super important.
And just as an anecdote, after this paper came out, I went to John November that basically wrote this paper, and I told him, okay, what happens if you have a mixed person? And then we came up with a different method that came up, 4 or 5 years later, we called it spatial. In any case, the idea over there is that you get 2 points for every person. And in principle, also 4 points, but we could.
Great. You get with the P1, just 1, 2. You have the, but it's just because these kind of studies are used to say, you see, these guys are not French.
Right. Oh, I see. Okay. No. Yeah.
It's they are French, but they are not like.
Yeah, and I'm not even getting into who is from which. That's not.
This is super important.
I'm sticking to the biology here. So, okay. So one way to think. So this actually doesn't work for methylation. So just kind of a preview. It doesn't work well for methylation. If you try to correct with PCA or something similar, it's just not going to work. One way to think about PCA. Kind of a probabilistic interpretation for PCA is that you have everything sits in a low dimensional space, let's say in 2 dimensions, and then you add normal noise. So in this case, the normal noise would be independent in all directions. So these kind of like balls. And then now you solve a maximum likelihood estimate in order, you can show mathematically the maximum likelihood estimate for this is going to be exactly the PCA solution. So it's not hard to show this, but it's useful to think about it this way because when you run PCA, you essentially do this. You solve that likelihood function, and that means you assume this. Okay, but that's not the right model because if you think about methylation, in methylation, that assumes that the noise goes in all directions. Yeah.
So when you say you're trying to use this to correct methylation, do you mean that you want to make the p-values that are hyperinflated more reasonable?
I want it to be, if I go back to this, I want it to be closer to that.
Yeah.
Because I expect that my hypothesis is that my assumption here is that most methylation sites are not going to be related.
Yeah. So my question is, does this affect the p-value effect? Does that persist if you just use like a permutation test-based approach, where you directly simulate the null given the actual observed data, which will account for the correlations and things?
But I mean, permutation test is tricky because it makes an assumption of how, depending how you do the permutation. You have to do the permutation stratified to the population, or stratified on the confounder. It's not so trivial to do the permutation in a way that captures so when you do a permutation test, you essentially define a null. So for example, if you just did a permutation, I'm just assuming that there's no confounders, which is not true. Let's say that you have two populations and you can do the permutation within each population, for example. Yeah. If you have a continuum or if you have another confounder, then it becomes much more, you could do the permutation test by, if you knew the confounder, the problem is we don't know the confounders. That's kind of like the issues that Elior mentioned earlier in his talk. If you knew the confounders, you could maybe run a regression model, and then use the regression model to decide on the probability for the permutation. And we have a paper from 20 years ago here with Mike Jordan on that we did something along these lines. But that you have to know all the confounders. Otherwise, it's not really clear how you do the permutation. But in principle, you're right. You could do this if you have a way to do the permutation test correctly.
I can ask you more maybe offline.
Okay.
Yeah, thank you.
Okay, so there is a theorem here. So this is one question for at least one problem for the theoreticians. So, and there's actually a missing guy here, which is Matan Gavish from the Hebrew University, that he helped us with this. The basic idea is that if you just think about it as a kind of in random matrix theory, then PCA basically assumes that you have a low rank matrix plus noise. And what we assume is that you have a low rank matrix that is defined on just a subset of the columns. You don't know which column. Here I just organize them on the left, plus noise. Okay?
Island, like a specific places where methylation is really dense and deep and highly methylated. Could you and leave the others? Let's say, would you separate this really something that looks almost? So
I don't know. Maybe you could use some kind of biology over there.
That's what I think.
Could be, I don't know. We have to try. We did try a bunch of things, like try removing some methylation sites with low variance and like things like. But it could be that something along these lines. But in any case, I think that was kind of an interesting discovery because that led to a kind of a very simple algorithm. That basically what you do is instead of this kind of sparse PCA algorithm, that what you do is you do SVD, so you take kind of a low rank approximation for your matrix, and then you correlate every column in the low rank matrix to the original matrix. Now you order the columns based on those correlations. The correlations that have high the columns that have high correlation are expected to be this. You can show this in simulations or actually mathematically, and then you just keep those and you ignore everything else. You make everything else 0. And then you run PCA on this. And that's basically that sparse PCA. we did that, and we proved that when reasonable assumptions, there's a kind of a theorem here that you could basically see that under asymptotically, this is going to give you better results than PCA always. I mean, if the truth is not going to be sparse, you don't lose. So you essentially get very similar results to PCA. So practically what it means is that what people should do, instead of just run PCA, first do this. You don't lose anything by just running SVD, keep low rank approximation, and then run these correlations, and then run PCA on the matrix after you zeroed out everything else. Now, there are some assumptions over there, and I can send you that write-up. But basically, so there is, I think, a lot of potentially interesting theory that could still be done here, because we kind of left it because it was a side thing for us. The theory was Matan came, and Matan is came from Stanford with like he wanted to do theory and random matrices, and so we worked on this a little bit. But I think there's a lot of potentially open problems here, how to choose the dimensions and things like this. So, and then in practice, basically, after you correct, you're left with these many sites that are related to rheumatoid arthritis. So that's a negative result. We basically took the 2013 paper that I was very excited about. We essentially showed that most of the sites are actually not related to rheumatoid arthritis after you do the correction correctly. Okay, so if we look at cell types in general, there is actually something that, again, Elior mentioned this very quickly this morning, but if you just, this picture here basically shows you an example of what happens if you have 3 cell types, in this kind of a toy example. The colors here say what's the methylation, how much is it methylated, and then this is the cell type composition for every individual. And what you get on the right side is what you actually observe, which is a mixture of, it's kind of a soup of all the cells together. But what you really want to do is this. You want to find the difference between cases and controls in each of the cell types. So what we were thinking, and we have a few approaches that are trying to do this, are how do we do the convolution and go from these back here, either completely and basically just not do, like you can do this experimentally and do cell sorting depending on the tissue, but let's say for blood you can do it, and that's going to be way more expensive, and it's another process. And in some tissues actually you can't really do this. And so, or you can try to do this in silico, basically in the computer, which is what we try to do. And I'm not going to go into all the details here, but I'm just going to say this. We start with a matrix of individuals by methylation. And then if we look at kind of PCA, non-negative matrix factorization, or any type of matrix decomposition that you're thinking of, you basically decompose it into the product of 2 matrices that are lower rank, okay? What we're saying is we're going to decompose it into a low rank matrix times, and now we have for every individual matrix. Now, you have to look at this and tell me why this cannot be just from an information theory perspective. That's impossible, right? So I know everybody's tired. It's after 40 hours of, so I'm not gonna, but just look at this. You start with, I don't know, n by m. Now you have n by k, and then k by m times n. You have way more data. You generate data. It's impossible, right, to generate, like we don't have enough information unless we make some assumptions. So I'm just going to try, maybe I'll skip this so that I'll be able to talk about one more thing. We have to solve this. I'll try to give you some intuition why that works. But to solve this, basically what we did is we put in place kind of a Bayesian model that makes us an assumption on how, kind of a generative assumption on how the data is generated. And essentially what the difference between the decomposition methods like PCA, non-negative matrix factorization, and so on, is that they all assume that every person basically has the same amount of methylation in every cell type, and the only difference is the differences in cell types. And we're assuming that there is a variable, it comes from a distribution. So the amount of, which kind of makes sense, right? I mean, but the thing is we don't know anything about the distribution. So the first paper that we had that, which we call TCA, and Lior mentioned it this morning, tensor composition analysis, assumed that there is a normality, and we ran it on the rheumatoid arthritis data, and we found 15 new methylation sites that we couldn't find if we just do a simple regression. And we actually replicated this on sorted cells. So this was actually replicated on sorted cells, and we could find the methylations, that these methylation sites are just differentially expressed or methylated in these sorted cells. Just quickly, why there's information here if you assume that you have, let's say, in this case, normality. So let's just say that you have 90% red cells, 10% blue cells, and you just assume that in both cases you have normal distribution, and actually with the same mean, same variance. So typically you can probably find out that they have different mean, that's not so hard, but let's just even say that they have the same mean, same variance. So, and then I see that I have methylation 0.7. So I can assume that both of them have 0.7, and then I can look at the density. It's just going to be 0.5 in each of them, so it's 0.5 square. It's going to give me 0.25. Or I can kind of shift one of them to the left, the other one to the right, and I can do it based on the 90% and the 10%, so that the total methylation is going to be still the 0.7, but then the density is actually going to grow. Okay, so because the reason is this one is going to be pushed way higher, okay, compared to this one. So the effect on this one is not going to be so bad. The effect on this one is going to be so great, right? So altogether, it's better to do it this way. So you have information by using this assumption. And we just recently had a paper just a few months ago that we published that basically does the same thing, but without the normality assumption, with the kind of non-parametric methods. I'm not going to go into the details here. Yeah, question?
This is really great because not using methylation because of this problem. So if you can solve the problem, that's remaining. But I thought that some people have found some signature, for example, of some cell types that they can use to.
Yes, right. It is true. So there are supervised approaches.
Okay. And
those would be, but there's a lot of limitations for these supervised approaches. And we can talk about them offline because I don't have time. The supervised approaches are going to help you to find the cell type composition. They're not going to be able to tell you the deconvolution, right? So, okay. I just want to talk a few minutes before I finish about going back to the methylation for the imputation. So again, if I go back to the problems with genetics, methylation solves some of these problems. Methylation is actually affected by the environment. We can figure out if someone is smoking, not smoking, if we can figure out the age. We can actually see if the mother of the person basically smoked when they were in the womb. Lots of things that on the environment we can know. Genetics is static, methylation is not static, and the third one is just, let's not talk about it because we spent 5 minutes on this, but basically there is the promise at least that we haven't done 20 years of this. So we still, in 2045, we can stop. So what we did here, and I'm just going to go quickly over this. So Dan Gershwin, by the way, is doing autism, but he's also in charge of the biobank in UCLA. And we went to the biobank, and so UCLA has a biobank with samples from patients that come to the system. We have the EHR data for them. We have the genetic data for them. And we generated methylation data for 1,000 patients in this case. So not a lot of, so was a bit more than 1,000. I think after we cleaned it, it was maybe 800, 900. So we removed some for quality control. And what you see here very quickly, I'll just go over one of them. So phi codes is basically the diagnosis codes, but they're just we have a few of them that are grouped together. So kind of if you broke your right arm or your left arm, it's still going to be the same code from. So these are groupings of the diagnosis codes. Every point here is going to be one condition. Just think about it as one condition, one diagnosis, and the different colors. One of them is the blue is the PRS. So this is using the genetics, and red is going to be. Sorry, I'm colorblind, but I think it's red. Is the methylation risk score. And you just generally see, and then the x-axis is basically how well do you do in terms of prediction when you look at the baseline. And the baseline here doesn't really matter, it's details, but it's using the kind of the normal thing that you think you should do for prediction, so age and sex and the first 10 PCs, right, or something like that. So, and then maybe cell type composition, I don't remember exactly. But basically, what you see here is that that's the baseline. And generally, the methylation risk scores is above the baseline. Generally, PRS is about the same thing as the baseline. That's true for medications. That's true also for labs. And I don't have time to go into all of this, but I could spend basically now 40 minutes just talking about all the results. But just generally, I would say, what we found, we compared it to 600 different phenotypes, including medication labs and phi codes, which is essentially diagnosis codes. And we saw that just generally, it's much better than what you get from genotype data. Definitely when we look at 1,000 patients, so the sample size of 1,000. We also went to the UK Biobank and we ran, we took the PRS from there and we applied it here. So UK Biobank has 500,000 people. And so you can run now a much bigger study to get basically the polygenic risk scores, and then you can apply it here. So you essentially give an advantage to the genetics. Still, the MRS were typically much better. So these are just some examples of the things that basically were pretty high.
Methylation from which tissues?
Blood. Yes, it's blood. But there are, if you look at the list of things that we saw that the relations, there were also things in the brain that are related. So things related to schizophrenia and things like this. Now, there's no causality, and there's no, it could be that someone is taking a medication that is related, or some, but we could find some people that have some conditions or take medications that are.
Let's say in biobank or whatever of the composition of cells, they do have like a measure for how much lymphocytes you have, how much.
Yeah, so cell type composition, yes, it's going to be that's going to be trivial here, right? But I mean, why would it be related to creatinine, right? So now it's clinically it's not very interesting that it's creatinine, but in principle, think about there's a measurement. These are the things that we know to measure. So as a community in medicine, we know what we know about what we need to measure. There's maybe something that we don't know that we need to measure. Maybe that something is going to give us two types of a disease, like we saw this morning with Elior. That there's my hypothesis is that what Elior found with the two subtypes could be basically something that we could potentially just find something else, which is not methylation, and that methylation just captures it. Yeah.
Sorry, very quick. So first, these are predictions, not imputations.
These are, I call them imputation because I predict the now. I don't predict what's going to happen in the future.
But it's not like a general sort of across historical data or something as well.
No. It's what people have right now. So the reason I call it imputation because I think about it at least.
No, that's fine. I just want to make sure I understood the structure.
It's just when people say prediction, typically in this context, they think about what's going to happen 3 months from now for the patient.
So call it estimation, not prediction.
It's semantic. It's fine. I can change the name. Yeah.
And my second question is, so the MRS and the PRS, is this a polygenic or a methylation risk score for one thing or for a collection of things? Like, how many numbers are in those features?
Each one of them is for one of the 600. So we trained a linear model essentially for each one of them. By the way, we tried methylation and genotype together.
Yeah.
Didn't give us more information to the methylation, but it's just a linear model for each of them.
Yeah, but so you have the risk score is for 600 different things. So you have a
Yes.
600 dimensional
Right,
vector
it's
that
not
you're using
foundational
for-
model at all. It's like
No,
very,
yeah.
so,
My follow-up question is, and I know this is technically challenging because of dimensionality,
Yeah.
and probably infeasible with a polygenic risk score, but with the methylation, you have 800,000 at the start. Maybe you could actually just go straight from the raw methylation data.
we do. I mean,
No, just directly to the thing you're imputing, not through the risk score itself.
No, that's what we do. Sorry, maybe I must.
Oh, you're not taking the methylation risk score.
We
and
take
then,
every phenotype, each one of the 600, and we just do a.
right, I understand.
It's
so it's
very
a separate predictor for each one of your results.
yes,
Okay.
yes.
Not a single methylation.
I basically I didn't do anything smart here from a computational perspective. What I did is just show that there's a lot of signal in methylation, and that instead of 1,000, we should look at 100,000, which is
Yeah.
the next thing we should do. But
One minute.
okay.
Huh?
Yeah.
Do you think that maybe the PRS underpowered because it was trained on a different population? Because it was trained on the...
I mean, again, to be fair, we only use 800 people. So if we like just apples to apples, the genotypes basically.
I don't think so. I mean, it's because the
General.
European, I mean, the GWAS were done on European and UKV are European, so it's.
Yeah, we did everything here is for like we have a few populations, but we took them out here for when we did the majority are Caucasians here. We actually show that. Anyways, I don't have time, but basically there's information also about the other ancestries. But yeah, that's probably not the reason. At least we don't. Okay, just one quick thing. We did one simple approach, which I think could potentially be improved. We just didn't have time to do more than that. We took the methylation risk scores, and we put that matrix next to the matrix of the EHR. And now we just plugged it into soft impute, which is a matrix completion. And it gave us better, in some cases very different results, but typically slightly better results than just doing the soft impute, which essentially, again, means that you can go to them, and this is without taking into account, soft impute doesn't take into account the fact that you have some distribution that you know about the methylation. There's a lot of things that you could, and there's kind of a missingness pattern here. There's a lot of things that you could probably take into account here to get even better results. But just as a way to show that you have something that methylation gives you more information, I'm going to skip this, but we have some results that show that instead of looking at the entire genome and doing a linear model, you can take 20 genes that are related to the condition and do a neural network on those. And then we don't get better results, but that basically shows you that you don't need the entire genome. You can just take the genes that are related to the disease and you'll get essentially the same results. I'm going to finish here, and for those of you who are interested offline, I can talk about the second potentially theory problem, but I'm over time.
Thank you so much. Question.
Yeah, I have millions of questions I would ask. First, I think methylation is also very useful. Sometimes there is a signature of a rare variant. So this is something that we are using more and more.
Mm-hmm.
So you don't see the variant, or you have a variance of unknown significance. You do a methylation array, and then you see the signature of methylation that, it looks like this variance is doing exactly what it is doing.
Yeah.
But this is super interesting. Do you have, from the methylation you found, can you go back to the methylated CPG and to see what kind of genes are involved?
I mean, you know where it is, but there's a few problems, correlations between the methylations. We work with the arrays, and so you're limited in terms of the data that you have. You don't have the entire methylation haplotype. We haven't done this work, but in principle, that kind of gives you a little bit of localization, and you can probably then just go and try to do more work.
And just you said a lot of things about the batch effect and the fact that you need to find a way to extract the. So, what kind of questions? Because I mean, if you take, it's so clear that the environment will change something. So, if you take the blood in the morning or in the afternoon, so what kind of data you can use to have an idea of how to reduce this?
Yeah, it's first of all, great question. I think we did try to answer some of this in the paper, and we looked at, for example, the time that you from the time that you have methylation until basically we looked at the measurement of the person, right? So you might have had, because these people went through surgery, so the methylation was taken before the surgery. So we looked at the time, we saw that actually didn't make any impact. We looked at a few of these approaches, but we can't really cover all of these things. I think to cover these things, we need to take methylation from people every hour. I think but that's going to be still expensive today, but maybe not expensive 10 years from now. I don't know. I mean, so this is because the prices are going down very quickly. We definitely need to take more than one. I mean, I think we should take more than one measurement per person. It doesn't have to be every hour. Right now, this is based only on one measurement. I mean, ideally, you should have one measurement and then another measurement a week later, or you know.
But if you have the time of sampling, you can try to see what kind of methylations are sensitive to.
You can do so many things, and I can talk for an hour about like how do you can find actually some of the biological confounders by looking at two different times, like what is biological versus basically what is technical. We have a work that basically does something along these lines. There's a lot of things you could do if you have more than one time point. It's just that we don't really have that data for most data sets.
Yeah.
But I think again with the reduced pricing and with like more examples that show basically that methylation is potentially useful, and I'm hoping that people will start generating this.
Okay, so thanks for all the survivors here. It was an amazing week. Really, we moved this week. Was really like a tour, amazing tour from theory, from formalization, from foundation models. I mean, really amazing, and quite a lot of biology and finding and discovering. So yeah, we can keep doing it over dinner.
And thanks to Etan.
Thank you so much.
And thanks to Etan.
Thanks
Yeah,
to Omer that organized the stage.
Because I'm the last one who survived.
Yeah.